Thursday, Friday, and that's what we do on Friday. We enjoy science. And some of you sometimes are stressed, sometimes by oxygen. And so to support that idea, to learn more about it, we have a fantastic speaker today. And I will just go to formal introduction. Tonia was trained by the best people in the field over many years, and also now contribute a great pieces of work in the last decade or so. So please, thank you, Tonia, for joining us.
Thank you, Chris, and UC Irvine for the invitation to speak. We have a student who's gonna.
Yes.
Okay.
Hello and good morning. My name is Hernan Gomez, and I am a second-year graduate student in Dr. Bochesky's lab here at the Center for Translational Vision Research at the University of California, Irvine. It is my pleasure to introduce today's seminar speaker, Dr. Tonia Rex. Dr. Rex completed a master's and PhD in molecular, cellular, and developmental biology at the University of California, Santa Barbara, exploring molecular and cellular effects of retinal detachment on the photoreceptors. She then completed her postdoctoral training at the University of Pennsylvania School of Medicine. Following this, she became an assistant professor in both the departments of ophthalmology and anatomy and neurobiology at the University of Tennessee Health Science Center. Following 5 years here, she moved to the Vanderbilt University School of Medicine, where she is currently a professor of ophthalmology and visual sciences at the Vanderbilt University School of Medicine, associate vice chair for translational research at the Vanderbilt Eye Institute, and the Marlene and Spencer Hayes Endowed Director in Translational Vision Research. With more than 100 peer-reviewed publications, chapters, and presentations, Dr. Rex has received many awards, including the Schaffer Prize from the Glaucoma Research Foundation, the Career Development Award for Research to Prevent Blindness, the Audacious Goals Challenge winner from NIH, and the Gold Fellow Award from the Association for Research in Vision and Ophthalmology. In addition to her many scientific achievements, she is a member of the Association for Research in Vision and Ophthalmology, the International Society for Eye Research, the American Society for Gene and Cell Therapy, and a member of the National Neurotrauma Society. Her current leadership roles include being the organizer of Works in Progress at the Vanderbilt Eye Institute, a member of the Women on Track Steering Committee, and chair of the Vanderbilt Eye Institute Outreach Committee. Her current work includes projects looking at mechanisms and therapy for visual system neurotrauma to understand the physiological basis for visual dysfunction, looking at erythropoietin-mediated antioxidant pathways in glaucoma to determine the role of the NRF2 pathway and antioxidant response elements in EPO R76E mediated protection of retinal ganglion cells, and also work investigating retinal ganglion cell replacement in clinically relevant models of optic neuropathy. Her collaborations include work exploring network mechanisms coupling closed head injury and pre-existing susceptibility to clinical and subclinical seizures, and also collaborations involving identification of subcellular first responders to traumatic brain injury associated injuries via high-speed nonlinear optical imaging. Without further ado, please welcome Dr. Tonia Rex.
Thank you very much. I love that you have a student doing the introductions. It's so hard to get up and speak, and we only get more comfortable with it by doing it more often. And thank you very much for that very kind introduction, and again for the opportunity to present. Can you all hear me fine? Yes. Great. Thank you. So yes, today I'm going to talk about oxidative stress. It's been an area of research of mine since I was an undergraduate, and talking about a couple of different optic neuropathies and how they are similar and dissimilar. I want to start by acknowledging the people in the lab who do the bulk of this work, and I've put in bold those who contributed to the projects that I'm going to talk about today, to include Alexandra Bernardo Colon, who is now working at the NIH, and Sarah Naguib, who is now a postdoc at Columbia, John Backstrom, and my collaborators, David Calkins, Craig Duvall, and Fiona Harrison, and of course, my funding agencies. Couldn't do any of this without support from the NEI, the Department of Defense, and private foundations and families. So there's two different mouse models of optic neuropathy that I work with the most. We have a model of indirect traumatic optic neuropathy, or ITON, because that's a mouthful. And we're interested in that because it is much more common than a direct traumatic optic neuropathy, which is typically modeled by optic nerve crush or exotomy. And it only occurs in association with traumatic brain injury. And it is more common in blast injury than in the civilian population. So it seems to occur in greater incidence in the military in that population. And there are no treatments for this condition that have been proven to be more efficacious than observation alone. And down below is our system that we use. So this is basically a paintball marker that we override the paintball mechanism. We can control pressure input to the system here and measure pressure output with a pressure transducer here. We can slide this out, place an anesthetized animal into this tube, and the machined barrel is the circumference of the mouse eye, as is the hole in the PVC pipe on the other side. So the air pressure wave only hits the mouse eye. That way we avoid a traumatic brain injury and just look at the effects of a blast on the eye and optic nerve. The other model we use is a model for glaucoma. Of course, we're interested in this because it's a leading cause of blindness worldwide. And the model we're using is a microbead occlusion model. It was developed by Rebecca Sappington and David Calkins. And it's nice because we can control the timing of the induction of elevated intraocular pressure. So we inject beads, they partially fill the trabecular meshwork and causes a moderate rise in IOP that I'll show you. So here is an example of this elevation of pressure that we achieve. It's sustained for about 4 weeks with a single injection of microbeads. This is a schematic from one of David Calkins's papers going over the general pathology in this model where we have axon degeneration that begins distal from the eye and progresses proximally. And then of course, there's also synapse elimination and other changes that have been detected. So we have a moderate but sustained elevation of pressure and a distal axonopathy. In ITON, in contrast, we also detected an elevation in pressure, but it was quite transient. So really after 3 days, it drops back and we're to normal levels. So it's not sustained enough to cause what we consider glaucoma. And when we looked at cross sections of the optic nerve, proximal, medial, and distal, and did so over time, we never saw evidence of a regional progression of the degeneration. It seems pretty consistent across the length of the optic nerve. So this is a moderate but transient elevation in IOP, and it's not a distal axonopathy. So these are not the same conditions. Also, in the microbead occlusion model, we know it's a slow progression of degeneration and vision loss, and we see that in these animal model as well. So if we look at 2 weeks after elevation of pressure, these are some examples of cross sections of optic nerves. You can see nice tight myelin sheaths and a clear axoplasm in a healthy, as well as in the mom optic nerve. And we have total number of axons that are comparable. But if we look at 5 weeks, now we have loss of axons. And down here, you can see some degenerative axons in this example, optic nerve, and see that increase in degenerative axons in the model. In contrast with the injury model, And this is a repeat exposure of a low pressure that causes this ITON. And when we do that, even at 2 weeks, you can see a pretty dramatic pathology in the optic nerve, a lot of degenerative axons, we have loss of axons, signs of Wallerian degeneration. And when we count total axons, there's significant loss by 2 weeks and a lot of degenerative axons still present at 2 weeks after injury. And you can see the time course here, where it's really the peak of degeneration is at 2 weeks and it really drops off. So an earlier, more rapid time frame of injury. What about visual deficits in these two models? Well, I have 5-week data in the microbead occlusion model, the glaucoma model, compared to 4-week data in the ITON model, but it's pretty close. You can see that the Bmax of the ERG, which is looking at those bipolar cells, are pretty comparable. And the Amax also is very normal in the glaucoma model at this time point. In contrast, in our ITON model, we published some years back showing that the Amax is normal in this repeat injury model, but there's a decrease in the Bmax. So again, a difference between these two optic neuropathies. What we showed in that paper was that this was due to synaptopathy. So there's retraction of the bipolar cell dendrites from the photoreceptors that occurs in this model. Similarly, if we look at the visually evoked potential, there's really no effect at 2 weeks. We have a decrease at 5 weeks, and that's quantified here in the glaucoma model. In the repeat injury model for ITON, there's a decrease at 2 weeks and a further decrease at 4 weeks, correlating with loss of axons. So just to summarize the pathologies between these two optic neuropathies, glaucoma is a distal axonopathy, ITON is not. Glaucoma, you have moderate and chronic elevation of IOP, whereas in ITON, it's moderate but transient. We have a slow axon degeneration in the glaucoma model, where it's more rapid in ITON. Functional deficits can precede the axon degeneration in the glaucoma model, including axon transport. And I'm going to show you some data with photopic negative response looking at ganglion cell function. And in contrast, the ITON, the functional deficits really correlate with axon degeneration. We published that in a Frontiers paper with axon transport in addition to the ERG and VEP. And then the B waves are normal at 5 weeks in the glaucoma model, but there's a decrease in ITON. RGC death appears to be a late event in both. But the real question is, are there any commonalities between these different pathologies on a molecular level where we could use a therapy for one that would work for both and really leverage that for clinical translation to help both populations? So let's look at what we already know. There were already papers showing in the DBA2J, this pigment dispersion model of glaucoma, that there is elevated lipid peroxidation that exists. Importantly, it even exists at 3 months prior to elevation of pressure. So there's something about this animal. The animal does cut, the retina does have increased antioxidant proteins compared to the C57 Black 6 control here. And there's also data from the rat episcleral vein occlusion model, which is a really high IOP model. And here again, you see increases in superoxide and lipid peroxidation and increased activity of antioxidant enzymes. So the question is, does this also happen in the microbead occlusion model? And second, and kind of more importantly, does this elevation in reactive oxygen species precede pathology or happen with the pathology? So we assess this through Dhe imaging. Dhe is a dye that signals for superoxide. It's a marker for superoxide. So wherever we see red here is an indicator of superoxide in the retina. These are images, in vivo images of the retina. And we quantified that to the right in this graph. And you can appreciate that even 1 week of IOP elevation, we have a significant increase in superoxide. It's even higher 2 weeks after, and then it drops, although still elevated from the saline controls, but it's not as elevated at 3 and 4 weeks, which is interesting. Why is it not as elevated at these time points? And then similarly, looking at nitrotyrosine, which is a byproduct of peroxynitrite's reactivity with tyrosine residues on proteins, we see this accumulation over time in our glaucoma model. So to understand why the levels of superoxide weren't as high at later time points, we investigated levels of antioxidant proteins, and in fact saw an elevation of mRNA for a variety of antioxidant proteins at particularly 2 weeks post-IOP elevation. And then that correlated with protein levels when we took a few select examples and ran Westerns, and in fact saw elevation of, say, PRDX6, GPX1, and superoxide dismutase 3 at weeks 1 and particularly at weeks 2. Now, how are those becoming activated? One really well-known pathway is the NRF2 ARE pathway. NRF2 is normally bound to KEAP1 in the cytoplasm and is targeted for ubiquitination, but under certain situations can be released from KEAP1 and translocate into the nucleus, where it binds to, along with MAF proteins, to the ARE, which is the antioxidant response element, this enhancer element upstream of a lot of antioxidant proteins, the transcripts for them. So we looked at NRF2 levels, and there was no change, which is fine. We don't have to have a change in total NRF2 protein levels. What was interesting is that we did have this large increase in phosphorylated NRF2 right at week 2. So the timing is making sense. We also looked at KEAP1. Its levels do not change, but there was a difference in the amount of NRF2 bound to KEAP1. There was a decrease there. So again, this is all matching up to activation of NRF2 being able to activate the ARE. So we next want to understand how NRF2 is being phosphorylated in glaucoma. And there are some pathways, potential pathways that are known. So we explored those. We found no changes in phosphorylation of JNK, ERK1, or GSK3 beta. However, we did see an increase in phosphorylated PI3 kinase and AKT. So the next step was to see if that pathway is particularly relevant for this endogenous antioxidant response to the elevation in IOP. And to do that, we treated with a couple of inhibitors to either PI3 kinase or AKT. The effects were the same regardless. So I'm just showing you the results from one of these inhibitors, in this case, warfarin. So when we treat with warfarin, we prevented the phosphorylation of NRF2. It also prevented the elevation in many of these antioxidant, an expression of many of these antioxidant proteins, and prevented the increases on the protein level as well. Further, if we knock out NRF2 using the NRF2 knockout mouse, we had increased superoxide. Importantly, the NRF2 knockout, even prior to elevation of pressure, had elevated levels of superoxide, and this really shouldn't be shocking, but it's important to recognize as we try to interpret the resulting data. So without NRF2, again, we don't have this elevation in antioxidant protein expression or on the protein level. Further, lack of NRF2 resulted in with elevation of pressure resulted in loss of axons even at 2 weeks after elevation of pressure, which again, we don't normally see. And here it's important to point out that the saline group did not have this drop. So this is a glaucoma effect. And there were more degenerative axons at 2 weeks. The VEP was reduced in both conditions here, and the photopic negative as well. So a bit of a confounder because of just the effect of lacking NRF2 in general. But it does suggest, since we get this earlier onset of axon degeneration in the absence of NRF2, it does suggest this endogenous antioxidant response is contributing to the delay of onset of glaucoma pathology, perhaps contributing to why it's such a delayed progressive slow disease. So we then injected these NRF2 knockout animals with an AAV intravitreally to deliver NRF2 to the ganglion cell neurons, primarily retinal ganglion cells. And then we induced elevation of IOP. And when we did that, we found that overexpression of NRF2 in those neurons was sufficient to overcome the global knockout and protect against elevation of superoxide. And we then once again saw elevation of these important antioxidant proteins. Also resulted in preservation of photopic negative response and the visually evoked potential and axons in the optic nerve. So that was pretty exciting. We then wanted to know, okay, what about with a healthy retina? Can we overexpress NRF2, say as a therapeutic and treat against glaucoma? And in fact, again, here, this is showing that when we deliver NRF2 via adeno-associated virus, we do get increased NRF2 as we'd expect. It's primarily in the ganglion cells, and it prevents elevation of superoxide that we would have gotten otherwise. We also get a further elevation in expression of many antioxidant genes and increased levels on the protein level for PRDX6, GPX1, and SOD3, again, just as example proteins. The photopic negative response in this treatment group was comparable to normal, as is the VEP, even out to 5 weeks post-IOP elevation. And the optic nerves were well preserved as well. Very little axon degeneration and good preservation of total axons. So just to summarize this part of the presentation, hopefully I've convinced you that reactive oxygen species are elevated prior to glaucoma pathogenesis. Elevation of pressure causes phosphorylation of the PI3 kinase AKT pathway that causes phosphorylation of NRF2, which activates the antioxidant response element, driving this increased levels of antioxidant proteins and delaying the onset of pathology. Lack of NRF2 prevented this response, causing earlier onset pathology, and AAV NRF2 gene therapy was protective. What about ITON? Well, the optic nerve is part of the brain, so let's look at traumatic brain injury. What's known there? There's a lot known there, and it's really quite evident that there's a significant role for oxidative stress in the pathology after neurotrauma, as there is a lot of contribution as well of inflammation, in particular the IL-1 pathway. And we've published on this as well in our ITON model, that we see activation of the IL-1 pathway and the inflammasome after injury. But let's look specifically in this model, make sure we're seeing it here. And yes, using DHE fluorescence again, comparing sham to our ITON model, you can see an elevation of superoxide at 2 and at 4 weeks after injury. And we also see this accumulation of nitrotyrosine that overlaps quite a bit with the retinal ganglion cells. Here's the IL-1 pathway data. We can see increased levels of IL-1 alpha. We see increased in cleaved caspase 1 at 4 weeks after injury, and we see an increase in IL-18 as well. We also see increases in IL-1 beta. I just don't have that on this particular slide. We then tried feeding the animals with a high antioxidant diet. So prior to injury, and then for the full course of the study, animals were fed high vitamin C and vitamin E. The vitamin C levels were given, this is low vitamin C diet, which was a comparator, and then our high vitamin C and vitamin E diet. We don't see any change in vitamin C levels because the vitamin C is being used to recycle vitamin E. And here you can see an increase in vitamin E in the diet of these animals. Correlatively, we did not see the elevation in DHE fluorescence in these animals. So there was not the increase in superoxide. This diet also prevented the increases in IL-1 alpha and IL-18 and the cleavage of caspase 1. So it blocked the activation of the inflammasome pathway, and in fact, it even prevented priming. So did it have an effect on histology? Yes. Similarly, so here is a high magnification image of part of an optic nerve from a sham animal, again showing these nice healthy axons and normal glia. 2 weeks after injury, you can see a lot of onioning, the loose myelin, whether it's trying to reform, degenerative axons, Wallerian degeneration. By 4 weeks, you can see glial hypertrophy, scarring, and loss of axons, and still some degeneration. But with this high antioxidant diet, there's a lot less axon degeneration present at both time points, a lot more axons present, and that comes out with the quantifications shown here. Here's the degenerative axons at 2 weeks we normally detect, but this high antioxidant diet, that does not happen. You see a little bit of a bump up here by 4 weeks, so we haven't completely blocked it, but it's very much suppressed and delayed. The visually evoked potential, we normally see this decrease over time in our injury model that was prevented with this antioxidant diet. So in the ITON model, then the secondary neurodegeneration, which is what we primarily study, is at least in part due to elevation of reactive oxygen species and activation of this inflammasome pathway, both of which are prevented by treatment with a high antioxidant diet. So let's investigate this a little further. It's great to be able to have a great diet, but we're not all really good about sticking to a really healthy diet. At least I am not. So investigating these mechanisms a little further, as was mentioned in my introduction, I've been studying erythropoietin for many years. And we have shown that this mutant form of erythropoietin is as effective as wild type. This is looking in the DBA2J animal, looking at the number of axons. We get equal preservation regardless of which form of EPO that we use. And using our mutant EPO, this is systemic administration of the AAV, but EPO is a type 1 cytokine. It's secreted and it can cross at low levels across the blood-brain barrier and the blood-retina barrier. And we do get increased expression of many antioxidant proteins here. So we wanted to know, is the neuroprotective effect that we're detecting with EPO due to this antioxidant effect, right? We know now that the retina has this endogenous antioxidant response. Is EPO feeding into that and pushing it further? And is that why we're getting neuroprotection? So really asking, does EPO cause phosphorylation of NRF2 and activation of the ARI? So going back to our microbead occlusion model here, I'm sorry for the pixelated view here, but the main point being that when we treat with EPO, it has no effect on IOP. It's going to be elevated regardless. So elevated intraocular pressure, our model as expected. We wanted to, in this case, rather doing systemic delivery of EPO, we wanted to make sure whatever effects we're detecting were truly from EPO acting on the retina. So this is intraocular delivery. EPO has a very short half-life, so delivering the protein would mean we'd have to re-administer fairly frequently. And to avoid the challenges of repeat intraocular injection, we instead packaged EPO into PLGA, which is now a gold standard delivery vehicle. So when we deliver PLGA alone, you can detect the low levels of endogenous EPO that are present in the retina. This is over 6 weeks. And here, when in our packaged PLGA particles, we can see that increased levels of EPO in the retina as it's been released from the particles. Looking at superoxide levels, again, you can see the PLGA alone particles in the microbead occlusion model, we have elevation of superoxide. Oops, go back. As I've shown you before, but not when we've treated with the EPO loaded particles. And here again is that data quantified. In fact, we do see increased phosphorylation of NRF2 in this treatment paradigm. Now, these particles were delivered a day after elevation of pressure, by the way. What's interesting is that we're getting phosphorylated NRF2 now 1 week after IOP elevation, whereas with the endogenous response, it wasn't happening until 2 weeks. So we get an earlier activation of the pathway. We do get increases on the gene expression level, the mRNA level of antioxidant proteins. This is just a couple of examples. Our other families are also increased. This is just to show you we detected it. And then we also use this reporter construct. So here we put the ARE from thyroidoxin upstream of TD tomato, and we transduce the retina with this AAV 2 weeks prior to elevating pressure, and then treated with either PLGA or PLGA carrying EPO, and look 2 weeks later. And here you can see that there's greater fluorescence in the animals that were treated with EPO. We have that quantified. I don't know why it's not on the slide, but hopefully you can see the greater red fluorescence here. So in fact, EPO is resulting in activation of the ARE. So just like with the endogenous response, we wanted to understand what the signaling pathways were, the upstream signaling pathways that were causing phosphorylation of NRF2. Like before, we tried all of these, and the only one that we saw phosphorylation of was ERK and GSK3 beta. So we then used an inhibitor of phosphorylation of ERK, which is this BIRB 796. And when we treat with that, it has no effect on elevation of pressure, as high as expected. Using that inhibitor prevented the preservation of the photopic negative response that we would have had otherwise. So here's the preservation of photopic negative response, and then with the inhibitor blocking the protective effect of EPO. It also prevented increases in a lot of these antioxidant proteins on the mRNA and the protein level. Further, when we look at the inhibitor group, it prevented preservation of the axons in our microbead occlusion model. So it's a different pathway than the endogenous response. Looking at visual function and preservation here, you can see that the treatment with these particles, even going out to 5 weeks, still maintain preserved visual function. VEP not quite as good as at 2 weeks, but still better than the control group, and total axons were preserved, and there's a lot less axon degeneration. The problem with this is that we have some toxicity. So, oops, I don't know why it's not showing up here. But looking at the values here, so the PLGA treated group, looking at the amplitude of the photopic negative response or the VEP, this is a much lower amplitude than what we would normally have achieved. I don't know why my little arrows aren't showing up, but really a healthy would be close to 60. So this value is not supra physiologic. This is kind of closer to normal, which means that EPO is having to overcome toxicity of PLGA. And we also saw it with increased levels of IL-1 pathway, some inflammation markers, and increased oxidative stress actually in the PLGA treated group. So we wanted to look at a better way to deliver EPO to the eye to prevent that toxic side effect. And we have Craig Duvall is a biomedical engineer here on campus, a faculty member there, and he had developed these really cool microspheres that scavenge hydrogen peroxide and other reactive oxygen species. And as they scavenge these, they break down. So I asked him if we could package EPO into these microspheres, and he said, I don't know. So we tried, and we could. So we purified our EPO, we packaged it in, and here you can see the release in vitro and in vivo. So these are released from our first generation particles. It's not as great as we had from the PLGA particles, but we were able to get some EPO in there and release it in the retina in vivo. When we tested this in the ITON models, going back to the injury model now, we tested these PPS particles and we tested the PLGA particles side by side. Here with the PLGA, you can see we have DHE fluorescence in our injury model and the PLGA alone particles, and it's quantified below. And that's prevented with particles containing EPO. Looking at the IL-1 pathway here, here's that increase in IL-1 alpha in our injury group that is prevented by treatment even with the particles alone. So even if we didn't load them with EPO, we prevented the elevation in IL-1 alpha. The PLGA alone group was worse than the saline injury group. So both of these got injury, but treating with PLGA on top increased levels of IL-1 alpha in the retina and in the optic nerve. Similar scenario for IL-1 beta, and that's true in the retina and in the optic nerve. Giving the PLGA carrying EPO did overcome that, but it meant that EPO had to overcome the toxicity of PLGA as well as the injury. Looking at the optic nerves again here, another example of a sham optic nerve. And here's after injury given saline, you can see the degenerative axons present. And then treating with PPS or the PPS carrying EPO, which looks the best. And this is compared to the PLGA alone or the PLGA group given EPO, and then the quantification of total axons down below. And you can see that there was improvement in the PLGA EPO group as well as in both PPS groups with or without EPO. Looking at visual function, we performed ERGs and VEPs here. And here you can see the reduction in the ERG Bmax in our saline and PLGA treatment groups, but less so in the groups receiving either PPS or PLGA containing EPO. The VEP is the only example where we saw further improvement with the PPS carrying EPO. So here we have better preservation of VEP. than the PPS alone group. And then you can see the PLGA group as well. So we then realized we needed to really optimize these microspheres and our EPO loading for ultimately going into glaucoma, which ironically is a more complex environment. So we increased the crystallinity of these spheres. And so now they hold their shape better. That allowed us to load more EPO into them. And we measured that through fluorescence. So we conjugated a rhodamine to our EPO and performed an imaging quantification of that. And here you can see in vitro and in vivo assessments of EPO loading and release. And you can see that EPO is released from these particles when you're in the presence of high levels of hydrogen peroxide. And this is the release over time in the retina with our new generation particles. So these are some of my most recent results. We have elevation of pressure regardless of the particles, as we'd expect. Down below is EPO release over time. So here are these new generations, our third generation particles, we call them PSES. And without EPO, you still detect endogenous levels of EPO. We're not sure what happened there. And then with EPO contained, you get this spike up and then gradual loss over time. So that by 56 days, all EPO has been released from the particles. Looking at reactive oxygen species levels, we use the Amplex red assay to quantify hydrogen peroxide in the retina after collection. So we're back to our glaucoma model here, use the microbead occlusion. And here you can see an increase in hydrogen peroxide in the PBS treated glaucomatous retinas. Single treatment with EPO was somewhat effective, but we had less and less hydrogen peroxide as we progress here to our optimal particles containing EPO. Looking at visual function and histology then, 6 weeks of IOP elevation resulted in good preservation of the photopic negative response and the visually evoked potential, and good preservation of the optic nerve. Looking at total axons, we have really nice preservation of axons, despite some ongoing axon degeneration. So it's not perfect, but it's very good. So in summary, hopefully I have shown you that ITON and glaucoma have different pathobiology, but they share a common molecular mechanism in oxidative stress. And that the retina really fights back against elevation of IOP and the induced reactive oxygen species through this NRF2 mediated antioxidant response. An overexpression or activation of NRF2, even in the ganglion cells, might be an effective therapeutic strategy. Also shown that EPO or EPOR76E is antioxidant through activation of this NRF2 pathway, but through a different signaling approach than the endogenous pathway, such that the two might be able to work in concert in a synergistic way. And it acts earlier when we treat with the microparticles delivering EPO. The microparticles appear to be safe and effective in both of these models of optic neuropathy. And we think that these third generation microspheres could be a good alternative to the PLGA particles currently used. And I think that's it for me. Yes. So thank you for your attention, and I look forward to answering questions.
So thank you very much. We will have a few questions from the web and from Zoom and local people here. So let's start with Jiang Yang.
Hi, Rex. I was just wondering the mutant EPO in terms of how does it act in the retina? Does it bind to a particular receptor in the retina or in the RGC? Did you look at the JAK-STAT pathway? That's supposed to be downstream of the epogen receptor. Thank you.
Thank you. We did look at JAK-STAT and we did not see changes, but you're absolutely right. And that's why it was one of the ones we looked at. We know that the EPO receptor is present in a lot of tissues, but at a low level, and it has a really high turnover at the membrane. And antibodies for it are challenging. There are not really any great receptors, antibodies for it. There's also evidence that it can, instead of the EPO receptor homodimerizing, it can heterodimerize. So this could be working through either of those pathways, and we've not explored that yet.
I have a good news for you today. The final comments after all questions will be asked, will be Sylvia today. Sylvia, get ready. Okay, Elizabeth. Okay, she's not there. Rafał.
Hi, Dr. Rex. I'm Rafał from Chris Polczewski Lab. That was an exciting talk, exciting results. But I was interested in this dietary intervention. If, for example, a subject would get injured, and then if you would introduce this vitamin E, vitamin C rich diet, if that would be helpful to prevent damage.
Right. Thank you. Thank you for the question. So I think what I understood is you're asking if the high antioxidant diet would be effective if given after injury.
Yes.
Yeah, that's a great question. We have not explicitly tried that, but the fact that giving the PPS antioxidant particles after injury was effective was a shock. I really didn't think they would be. I thought the cat would be out of the bag. There would be so much inflammation that it really wouldn't work. So that surprised me. So maybe, that high antioxidant diet would be. I guess it depends on how quickly and how much vitamin C and vitamin E was being taken. Because certainly most of us, even though we're not scorbutic, we're probably taking the FDA recommended amounts of vitamin C. We do not saturate our receptors typically. So our brain is typically not saturated with, we're not receiving as much vitamin C as it could.
Thank you. Okay, we're going to Dominic and then to Dorota.
Okay, hello, Dr. Rex. This is Dominic from Chris's lab. I will continue on Rafał's question, but more of a technical question. So in what form of vitamin C and vitamin E you use? And how did you monitor the dose that animal took from the diet? Like, did you, so what was the dose per kilogram per day per mouse? And second question, how would you translate it to humans? Taking into account that they have much slower metabolism, how would you translate those to humans potentially? Thank
right. Those are great questions. That project was in collaboration with Dr. Fiona Harrison, who's a faculty member here, and that is her entire focus of her work is really investigating vitamin C and its roles in neurodegenerations. So you hit on something that I didn't bring up in my talk, which is that mice can make their own vitamin C. Sometimes it's good to be a mouse. So we could not use wild type. Well, we could. You can certainly use wild type mice, and we did. But we're supplementing on top of them already making a lot of vitamin C. So with the low vitamin C diet that we also tried, and I didn't talk a lot about it, we had to use glow knockout mice. So we had to remove the enzyme that allows them to make their own endogenous vitamin C. And that allowed us to really manipulate levels and look at the role of vitamin C in the pathology. So to get at your question about the amounts, it wouldn't translate to humans because we're treating wild-type mice that make their own vitamin C, and then we're giving above and beyond vitamin C and vitamin E. So it would have to be different for people understanding our own physiology and biochemistry. So it's a good question.
Hi, Tonia. Wonderful talk, and I really enjoyed the new data. So it's beautiful spheres, and possibly maybe something will be about that. I have a question which is related to age. So did you check for NRF levels or phospho NRF levels in older animals? And given that that there is possibly, I would bet, a higher level of oxidative species in the older animals, would you expect to have effect problems later, earlier, and then how it would prevent your treatment? How would prevent? What do you think?
Thank you. Thank you for your comments and for the great question. Absolutely, right? Glaucoma is age-related neurodegeneration. Neurotrauma in elderly population can be more devastating. So certainly the age is contributing. I would fully expect, I mean, from the data that exists, the oxidative state in older population is higher. So, you know, there's more ROS present. So probably if we exacerbated that or increasing the levels even further. And so, you know, I would expect it to be worse. We have not performed the experiments to date. And I'm sure you hear that a lot. I'm sure you're trying to encourage all of us to do this. And I appreciate that because it's important to do. We started here. I wanted to really understand mechanism, you know, what was driving the pathology. So we're really excited to identify this endogenous response of the retina, seeing it fight, you know, it was really, really cool. So that is definitely what we want to do, is test this in older animals.
So just following up a quick comment, you can probably do the longitudinal studies when you give your either diet or spheres or something for longer time, and then test the group whether this one will be now protected.
That's right.
Dr. Winkler. Lee. Wake up, Dr. Lee.
Hi. Hi. Very nice work. I have two quick questions. First, how is the where does the receptor, if you will, that sense the elevated IOP lie in the retina? Is it the RGC cell body? Is it the nerve fiber layer, or the lamina cribrosa, the exit point? And the second question is, in normal tension glaucoma, do you still expect to see similar elevation in oxidative stress? And if so, what might be the trigger for that? Thank you.
Thank you. Those are great questions and active areas of investigation for a number of laboratories. I think the answer is yes. We know that there's mechanosensitive receptors in multiple cell types in the optic nerve head and in the retina, and their roles in glaucoma pathology are still under investigation. So there's no clear specific answer to that I could say that we've identified. It's clearly TRPV4 or something like that. I think it's more complicated than that, but clearly they're playing a role. In terms of normal tension glaucoma, some would argue that it's a misnomer, that even in a lot of normal tension glaucoma patients, giving them IOP lowering drugs results in a reduction of pathology, a slowing of progression, suggesting they're responsive. So it's more of a sensitivity to pressure. On the other hand, there are genetic models now available for research, OptiNeuron being one, which is kind of this normal tension glaucoma model. And that is something we are very curious to investigate. So it's definitely on our radar. So thank you.
Hey, Dr. Winkler, do you have now connection? John.
Oh, yeah.
Hello, Dr. Rex. Great presentation. I was just wondering, just wanted to learn more about the mechanism of the axonal damage. Is that the mechanism that you described of the RGC damage to the RGC that causes the axonal regression, or is it something with the ocular anatomy or orbit itself that causes the damage, mechanical damage to the axon first, followed by the RGCs, or in this blunt force trauma or glaucoma model that you're presenting? Is it some sort of mechanical, or is it more of a cellular insult first, followed by an axonal degeneration?
Yeah, no, it's a really good question. The retinal ganglion cell death is a late event. Axon degeneration occurs first, so it's an axonopathy. So not that there are no changes at the ganglion cell level, certainly in glaucoma, we're seeing synaptic, dendritic retraction and some alterations in the physiology, but the axon degeneration seems to precede. So the question of physical versus molecular is an interesting one. With the IOP still elevated, we're getting protection. So whatever physical effect is happening, addressing the molecular changes are sufficient to retain structure and function. It kind of goes back to the previous question about these the responses, these receptors, right? So a physical effect will have a molecular effect. They're kind of paired. So I fully, and we've seen that with some of the work I'm doing. It was mentioned in my introduction, collaborating with some imaging engineers, looking at initial events after injury, for example. And this isn't glaucoma per se, but this is the injury model. And what we see right away is these increases in calcium, which isn't surprising, but it means that the injury itself, that push, is causing a molecular change very rapidly, right? So I know I'm not really answering your question, but that's kind of the state of the knowledge.
Let's go to Alan and Chris.
Hi, thanks for a very stimulating talk. That was exciting to see. So as you point out, mice make their own vitamin C. So humans probably top out at about 140 milligrams a day in terms of saturating. So it's not that much higher than the RDA. But have you tried like things like, you have glutathione as an extremely potent antioxidant, right? So have you tried things like alpha lipoic acid and nicotinamide, things like that, that could really stimulate dosage? Because your thyroidoxin data would imply that there's room there, right?
Yeah. No, you're absolutely right. And David Calkins with Denise Inman, they tried alpha lipoic acid in the glaucoma model and the DBA2J, and it was effective there. So I fully expect it to also be effective in the microbead occlusion model. You're absolutely right about glutathione. So yeah, there's plenty of work that we could go, direction we could take this. But yes, no, you're spot on with alpha lipoic acid.
Thanks.
Thank you.
You're completely muted.
I'd like to follow up on Don Minkler's question. And he had asked about whether mitochondrial accumulation is going to affect the oxidative stress. And have you looked at the mitochondria and what's happening in your model?
Right. It's not something my laboratory is equipped to do. So I would need to collaborate with somebody, and I'm very open to doing that. Clearly, that is going to be the source of the superoxide. It's got to be coming from mitochondrial dysfunction. It's the most likely source. So we need to investigate that. I fully expect it as a culprit.
Okay, well, I might contact you. Thank you.
Yes, please do.
Okay.
All right, we had a wonderful conversation. Let's go to Emma and then Anand, and then final word will come from Sylvia.
Hi, Dr. Rex. I'm Emma, and I'm a postdoc fellow here at the CTBR, and I work on Diabetic retinopathy. And I know that in Diabetic retinopathy, there's a lot of oxidative stress going on in the retina. And recently there's a clinical trial that is ongoing where they are given a non-steroid anti-inflammatory drugs, ketorolac, to decrease that oxidative stress by NRF2. I was wondering if you have done any comparative studies with your drug and versus non-steroid anti-inflammatory drugs.
Did we get a response yet?
Right, so thank you for the question. I think what I heard is you're asking if you're talking about a study being done in diabetic retinopathy using steroid approaches and I think
non-steroid
anti-inflammatory drugs. And I know that there are clinical studies using NRF2 activators. And it's interesting. I would have thought that could be a really promising approach, but correct me if I'm wrong, I'm hearing there's some pretty negative side effects to those activators. And, you know, I tend to be kind of cautious, and the idea of long-term activation or overexpression of a transcription factor kind of gives me the willies. I mean, maybe it'll be really effective, but NRF2 drives, you know, when it acts on ARE, the ARE drives expression of a lot of different things. So whether we want that to happen long-term, what kind of side effects that would have, you know, is I think something we need to think about. And it's one reason I really like in terms of EPO, you know, as I think about trauma, not delivering that long-term. So that's why I switched from AAV to these microparticles. So it's not permanent and it will stop. And I like that safety feature for this application. So I don't have an answer in terms of your particular study, but diabetic retinopathy, certainly I think an antioxidant approach would be attractive. EPO gets complicated in terms of its potential role in interaction with VEGF and neovascular changes and all of that, but activating the ARE in a short time frame might be useful.
Yeah, Tonia, very nice talk, very intriguing as well. I think oxidative stress has been implicated in sort of almost everything, whether it is a progressive disease, a long-term disease like AMD or glaucoma, or whether it is any other acute model. And as you pointed out, the two models that you're studying are giving you a pathology, but they're acute models. You know, there is an immediate injury, whereas glaucoma in general is sort of a long-term chronic stress. There are two related questions here. One, whether the mechanisms that lead to stress in glaucoma may be caused through different mechanisms. And in your case, you are looking after several days of injury, a week, I think. And is it possible that the acute trauma or injury initiates a different cascade of events that eventually leads to oxidative stress, which would mean that oxidative stress is not the primary cause, but a secondary manifestation of injury. Whereas in aging or glaucoma or AMD or other models, oxidative stress is again a secondary, tertiary, maybe quaternary sort of consequence caused by chronic sort of stress conditions and maybe many other genetic, epigenetic, or some other changes. You know what I'm saying? I mean, is it possible that you are reaching the same conclusion, but you're reaching from two different paths?
Mm-hmm. Right. It's complicated, isn't it? And I actually really enjoy studying complex neurodegenerations. We will never be bored or out of jobs. But I wanted to look early to understand what was driving the pathology, but we did look out as far as 5 weeks. Now, what we haven't done, and this kind of gets to Dorota's questions too, is, what about delayed treatment, right? What if we wait until glaucoma has progressed to the point where now you have different parts of the retina in different stages, and you've got some later stage occurring with the earlier onsets, it's a segmental degeneration. And now we try to treat with this antioxidant approach, it may be less effective. And that's kind of what we thought after trauma too. We thought the cat would already be out of the bag. We would have all this inflammation and all of this. So those experiments are going to need to be done. Will delayed treatment be as effective? And certainly,
I was actually thinking of early experiments. I was actually thinking, why not check immediately after trauma over the next 1 to 5 days and see what the mechanism is which is leading to oxidative stress. And could that have some relevance to the generation of glaucoma, which happens with age?
No, that's a great question. We did start some of those studies. So I do have some data at 1 day, 3 days, some earlier time point data. We just don't have the N as of right now to present on that. But you're right. And then that's also some of the work where we're looking actually seconds after injury. What are those primary events, primary changes, seconds to minutes after injury. So that's another set of experiments that we are working on. I just don't have that to present just yet.
Maybe a phosphoproteome study at early stage.
Yeah,
or
now that we are, that actually is one where, we have phosphoproteome data. So that's exactly what we did. Yeah.
Unfortunately, we will delay Sylvia for one more question from Lakshmi. Lakshmi, are you ready?
Yeah, hi. I have a question about the model. So does the optic of degeneration in the microbead model continue even after 5 weeks, after the IOP comes back to normal? Is the stress still there?
That's a great question. So as the IOP, if we were not to re-inject with microbeads and if we were to allow the IOP to return to normal, the understanding is that the pathology would slow because the inducer is no longer there. Just like when patients take eye drops, it slows the progression.
Yeah.
So typically, but if we keep re-injecting with the microbeads and we keep that IOP elevated, then the progression will continue for as long as we want to continue the animal.
Okay, so now highly anticipated final comments coming from Sylvia.
So it's nice to know that I was just hired by Chris. I'll send you my account number a little later. Hi, Tonia, this was such an awesome talk. Thank you so much. All my questions were pretty much already asked, especially the aging question, also the diet question. So from a mouse surgeon type of perspective, one other thing that came to my mind was in the oxidative stress models in the outer retina, we see a lot of strain differences. I mean, not just pigmentation, but even just 129 versus black 6, etc. So my first question was almost like a mouse to mouse research question is that also the case in your types of studies? And my other question was, it seems like you're looking very much at a cell autonomous effect almost on the RGCs, which is quite surprising to me. And so I know in response to Anand, you just briefly mentioned inflammation, but I was wondering if even in this very acute phase that you're looking at, if you see a lot of involvement and activation of Mueller and microglia, and if any of your treatments would also be enhanced if at the same time you would somehow subdue these types of cells. Thanks.
These are awesome questions, and congratulations, Sylvia. I hope you're very happy in your new location. Sounds like a fantastic place to be. So yeah, in terms of mouse strains, absolutely. We not with the repeat blast, but with the single hit injury, we compared black 6 to bulb C to the DBA2J, and the pathology phenotype is actually different across strains. And it, you think the bulb C, right? We know it's more susceptible to octaner crush. when we do blast exposure, a single hit causes optic nerve degeneration in that strain, but not in the black 6. RPE pathology is actually much worse in the DBA2J than any of the others. So yes, there are definitely strain differences, and the phenotype that you see can kind of matches what is known in the literature and what's known in terms of the differences in those strains. The pathology and the RPE that we get from the D2 with this blast, it's vacuoles. It looks very much like a lot of oxidative stress in the RPE. So we have not repeated those studies with our ITON model, this repeat hit. But that's a great point. I could certainly do that. I fully expect to see some differences based on strains, and I expect oxidative stress to be there regardless, because as an onset, it's always there. So, cell autonomous, yeah. So we wanted to investigate that because the retinal ganglion cells don't act in a vacuum. Those glia are close by and certainly contributing into the kind of the course of glaucoma. And so we obtained NRF2 flox mice. I just didn't have time to present this today, but we obtained NRF2 flox mice, and we treated them either with a glial-directed AAV driving CRE or an RGC-directed AAV with CRE, in order to begin to understand the roles of these two cell types in this endogenous antioxidant response and the pathology. It was not a clear-cut answer. You know, there wasn't black and white that, oh, you know, the RGCs are entirely driving this process and the glia aren't involved, or vice versa. That wasn't what the results showed. And frankly, you know, if we think about the biology, it makes sense. We know that these cells are communicating with each other and pretty extensively. And so it's going to take more work and probably some, you know, there's limitations. As much as I love to work in vivo because everything is there, to understand this level of interaction and mechanism, we're going to have to take it out of the animal and do some cell culture level studies. But it's a great question. It's something we are trying to pull out. So, you know, it was surprising that overexpressing NRF2 just in the ganglion cells was sufficient. I mean, these cells are injured, they're hurting, and yet they were able to treat themselves in a sense. I don't know that I would have expected that. So the glia are definitely playing a role. We know when we treat with EPO, treating either the glia or the neurons, EPO can act on them and acts different ways sometimes on those cells. So it's not to say that the glia aren't playing a role, but it's going to take more work to tease out the differences. I hope I answered all your questions, Sylvia.
Super, thanks so much.
Thank you so much, everyone. This was a wonderful presentation and great discussion. And I have a news for you. Next Friday at 8:30. I wanted to see you there.
Thank you very much. Thank you.